Hello, my name is Daniel Core, and today I'll be explaining one type of epistasis. Epistasis is when two different loci create the same phenotype. My example today, we're going to use single recessive epistasis. This type of epistasis is when homozygous recessive conditions at one locus mask the expression of alleles at the second locus. The example I'm using today is the pattern on the heads of snakes. The three phenotypes are plain versus split versus the double split. Dominant alleles are the big A and the big B, and the recessive are the little a and the little b. An image to better understand this, we have the plain, split, and the double split. The expression of a dominant allele of A allows for the expression of split, and then also with the dominant expression of B allows for the double split. Any expression of a homozygous recessive gene stops the expression of the second one. So at the first stage, if there is a homozygous expression of A, it doesn't matter whether the B expresses recessive or dominant alleles, it's going to stay plain. If it expresses a single dominant A, it can express the split. And if it expresses homozygous recessive B, it's going to only exhibit split. And then to be double split, it has to express both A and B enzymes, which are the dominant alleles. In a stereotypical dihybrid cross, it's typically 4 phenotypes are expressed at a 9:3:3:1 ratio. When single recessive epistasis occurs, at A, there's a 3/4 chance of expressing the dominant A, while there is a 1/4 chance of expressing homozygous recessive A. Just from that 1/4 expression, those will be the ones that will stay and only express the phenotype of plain. The 3/4 that move on to look at the B will have a 3/4, 1/4 split of expressing dominant B versus homozygous recessive B. The 3/4 that express this enzyme B, the dominant allele, would move on to express the double split. That 1/4 expression are the plain, and then 3/4 that move on to the intermediate times the 3/4 that move on to the double split would be the 9/16 expression chance of being double split. This intermediate stage, 3/4 have a chance to move to the intermediate stage, and 1/4 of those will express homozygous recessive and be the intermediate stage, which is the split, which this results in a 9:3:4 ratio, which you can see is different and doesn't have that 4th phenotypic expression. Another example is this one here. And when you look here in the beginning, there is a 50-50 chance of whether or not they express dominant A versus homozygous recessive. Just this right here, 50% of offspring from this cross will result in the plain phenotype of the snake head expression, while the other 50% move on and have the chance to express the B enzyme. This cross results in all offspring having at least one of the dominant alleles. This means all 4 would move from the intermediate state to the final one, which is the double split. This one, they do not express 4 phenotypes, but only express 2, as none stay in the intermediate state because there's no expression of recessive B. Thanks for watching.